Hello, everyone. Today, we'll be deep diving into the WHO Q1F Stability Guideline published in 2018, a critical update that influences how we develop, register, and monitor the stability of active pharmaceutical ingredients and finished pharmaceutical products, APIs and FPPs. This guideline replaces the older 2009 version and responds to global needs, especially in climatic zones 3 and 4, where environmental conditions like heat and humidity challenge product integrity. Whether you're in QA, RA, manufacturing, or validation, understanding this guideline is vital for ensuring compliance and maintaining patient safety across markets. Let's begin with a bit of background. In 2009, WHO issued stability guidance aligned with ICH Q1F, covering climatic zones 3 and 4. These zones include many countries in Africa, Asia, and Latin America where heat and humidity are high. However, the ICH later withdrew Q1F due to inconsistencies in global adoption. Some countries wanted a higher safety margin, more stress testing, longer durations. WHO then surveyed member states, resulting in consensus around 30 degrees Celsius per 75% RH as a long-term testing condition for hot and humid regions. This guideline became essential for product registration in non-ICH regions and is now the global benchmark for tropical stability. The objective of this guideline is simple but powerful. Define a core stability data package required for registering both new and existing APIs and FPPs. This includes not only first-time submissions, but also re-registrations and post-approval changes. It applies across the board, brand, generic, and in most cases, biologicals, though biologics have added requirements under ICH Q5C. The key takeaway here is whether you're introducing a new molecule or modifying a legacy product, the same scientific and regulatory rigor. When it comes to API stability, the goal is to understand how your molecule degrades and under what conditions. That starts with stress testing, heat, humidity, oxidation, light, and pH variation. Why? Because degradation products can impact safety and efficacy, and your test methods need to catch them. You must test a minimum of 3 primary batches, produced using the same synthesis route and packaging system intended for commercial production. Test specs should include all stability indicating attributes, chemical purity, assay, physical characteristics, and if applicable, microbiological integrity. These aren't just checkboxes. They're a defense against failure later. Now, on to storage. You need to test APIs under long-term and accelerated conditions appropriate for the market. Long-term, 25 degrees Celsius per 60% RH, 30 degrees Celsius per 65% RH, or 30 degrees Celsius per 75% RH. Accelerated, 40 degrees Celsius per 75% RH for 6 months. If there's a significant change during accelerated testing, meaning the API fails spec, you must conduct intermediate testing at 30 degrees Celsius per 65% RH. For APIs stored in refrigerators or freezers, specific conditions apply. 5 degrees Celsius plus or minus 3 degrees Celsius and minus 20 degrees Celsius plus or minus 5 degrees Celsius, respectively. This isn't just science. It's about protecting the supply chain from heat excursions during storage. Finished pharmaceutical products bring added complexity. Your stability study must cover all strengths of the product, all dosage forms, and packaging configurations. Unless you're using bracketing or matrixing, and that needs justification per ICH Q1D. FPPs also require stress testing, including freeze, thaw studies for liquids, photostability, hold time for bulk or in-use conditions. Also remember, the shelf life specification can differ from the release specification, but only with solid justification. typically for preservatives or assay drift. This is where the numbers talk. You analyze degradation using regression analysis and confidence intervals. The goal is to predict when your product will no longer meet acceptance criteria. You can use extrapolation beyond observed data, but only if the degradation model, batch variability, and mechanism of degradation are well understood. As for labeling, use precise, justified language like store below 30 degrees Celsius. Avoid vague terms like ambient temperature, they're non-compliant. Indicate special conditions if freezing is prohibited or reconstitution alters shelf life. Your label must speak the truth. This is where many companies stumble. Any post-approval change in formulation, process, pack size, even site can impact stability. Who requires that you submit updated stability data where appropriate? Commit to placing at least 3 production batches into long-term and accelerated stability. Maintain an ongoing stability program, 1 batch per year per strength and pack. Remember, change control without follow-through is a compliance risk. Stability isn't a one-and-done activity. It's a living commitment throughout a product's life cycle. In conclusion, this guideline is not just about checking boxes. It's about ensuring real-world robustness for products intended for some of the most challenging climates on the planet. If you're working in quality assurance, regulatory, or validation, I encourage you to not just understand this guideline, own it. Your ability to interpret and apply stability data directly impacts patient safety and global access to medicines. Thank you, and I'm happy to take any questions.